So now we are going to start the first session with the theme of diseases and One Health. We have three presentations with 30 minutes each. The first speaker is Dr. Salman, who is a professor in the Department of Clinical Science and is founder and director of the Animal Population Health Institute of College of Veterinary Medicine and Biomedical Sciences at Colorado State University. He is a diplomate of American College of Veterinary Preventive Medicine and a fellow of American College of Epidemiology. For more than 30 years, Dr. Salman has been involved in training sessions and workshops, both nationally and internationally, in the field of epidemiology, surveillance, disease management, and risk assessment. Please join me to welcome Dr. Salman with big hands and show your smiling face too. Dr. Salman, could you share your file and start the presentation? Dr. Salman, can you hear me? Yeah, your file is shared with us, but I cannot hear your voice.
It's my fault. I was on mute. So sorry for that.
No, that's
fine. Do you
fine.
see the file?
Now it's working, so you can start.
Okay. Well, I really appreciate the introduction, and I much appreciated the invitation by the organizing committee of this symposium. And it's my pleasure to share with you whatever I have for today as per the request on the topic I will present. I thought like maybe the best thing is to show the link between One Health concept and biosecurity, and with some philosophical and historical background. So to give an idea and appreciation for this link, but also to the One Health concept. I should start with a wonderful physician and philosopher, a German philosopher, Rudolf Virchow, and you could see from the old days, what he said is between animal and human medicine, there is no dividing line, or nor should be. And the object is different, but the experience obtained constitutes the basis for all medicine. I think that is really a good core for what we like to call the one health on one medicine. Little bit about the history of that is the One Health or one medicine actually started to be used by another gentleman, William Osler, who was a student of this physician, and he was a Canadian physician. And by the way, even though he's a physician, he started the first vet school in Northern America, in Quebec at that time. And also he was a very good pathologist, but his father was a butcher. So he learned from his father how to link between pathology and animals and human medicine altogether. Another, I would say, important person in the issue about one medicine or one health is Daniel Salman, who was the first veterinarian to start what is called at that time the Animal Health Bureau in the United States. And of course, you recognize from his last name, he was the one who named the bacteria, is now we know it has the common host between animals and humans is the Salmonella species. Dr. Salman, what he did in that time, and he really continued during his career is to link the health events to host, and the host here, whether it's animals or humans, but he kept in his mind the most important thing is the public health aspect or the human health in that way. Another person who really contribute to that is the Theobold Smith. He was not even let's call it medical profession, or neither a veterinarian nor a physician. He was a parasitologist, but he really worked on blood parasites in which he didn't even have the idea he was working on blood parasites, because he linked them to the ticks at that time, and the ticks, how they are associated with cattle, and how it led to a lot of the work and the early work on the contagious bovine chloropneumonia, as well as other diseases in that aspect. I like to call these are leaders in public health, is not in veterinary medicine, is not in human medicine, in the public health in general in that respect. So currently, and you heard a lot about one medicine and one health, and there are so many definitions and interpretation for this concept, and especially during the last few years, and particularly during the pandemic now we are going through. And I will try to emphasize on the link between One Health and biosecurity, and also how it's related to the COVID-19 as we go through it. But before I will start with that, I like to have a wonderful, I like to call it a definition of One Health, that it came from a committee by the American Veterinary Medical Association in 2008. And it's mainly emphasized on the cooperative efforts of multiple disciplines. And that's where is the previous speakers, they emphasized on the multiple disciplines, working locally and nationally and globally to attend optimal health of people, animals, and our environment. I really think that is saying all what we need to say about that. However, what I like to say is, when you look on the profession of veterinary medicine, what else we do? Actually, that definition of One Health covers everything, in my opinion, what we do in a comprehensive way in veterinary medicine. So my next slide is to show what's the link between animal health and human medicine. I like to say is, and I give a credit to the human medicine, specifically public health, for the initiation and the foundation of the veterinary medicine. Because if you look on the mission of or historically, but also even today, is there should be the benefit to human health, whether it's through the food safety, or whether it's through environmental health, or whether it's through even the quality of our life through our companion animals. So that all of it is to say, well, even though we care about animals, but is our ultimate goal is the human health in that respect. So that, I think it's a very important historical background in that way. During the 20th century, and is extended even to the beginning of the current millennium, is cooperation between these two disciplines, or between these two topics, human medicine and veterinary medicine, became less or diminished, mainly for several factors, and I will not go through all the factors. Some of them are legitimate, some of them are not, but because of that, the gap between these two, I would like to call them the medical professions, started to have a really separation, until, in my opinion, where COVID-19 came in the picture, which I hope it will be at least part of the, let's call it like the future direction will link all of us together in another way. So one thing started in the early or late 70s and early 80s is, and I'm proud to say, is Dr. Calvin Shuabi, my mentor, had a good vision about the veterinary education by focusing on agricultural production and population medicine, as he called it, preventive veterinary medicine in that respect. By doing that, he already started to initiate some tools and approach to preventive veterinary medicine using a population base, but in certain time is like also related to the agricultural production, which participated in some way, be extending the gap between the human medicine and veterinary medicine, because production or agricultural production, the human medicine, they said that's not part of our interest in that way. However, he did not mean it like to deviate from the main issue in that respect. And I will leave it up to here as far as like what is the intention and the pros and cons in that respect. Let me move a little bit about the biosecurity. And I want to start is here, I am talking about biosecurity and not only biosafety in that respect. The way I defined our biosecurity is all hygiene efforts designed to reduce the risk. And I use this as far as like for mainly in the veterinary medicine, but I can tell you it's applied also on the human side, and particularly now when we are going with the COVID-19. I presented this definition as well as the rest of the slides related to biosecurity in 2006. I did not change a lot of these wordings, but I will try to emphasize on the link to these with the current environment, what we are observing now with the current pandemic and how we are really dealing with it, both in good and bad part in that respect. So what biosecurity means is that it's more than just the quarantine or isolation. And by the way, I want to remind all of the participants, before January 2020, quarantine as a word was not so much used as compared to after 2020 and currently because of the COVID-19. However, the word quarantine was used way back in the last century because of the harmful effect of some of the human pathogens. Quarantine did not stop and used and applied in veterinary medicine during all the time and with all the good or the bad part about it in that respect. But I also want to say is the application of biosecurity is more than what we do in a clinical setup or on a premises level. It can be applied on regional, it can be applied on a national, but also anything between those, it will have that issue. There are more challenges related to veterinary facilities than in the human ones. And like biosecurity, for example, which can be applied in human hospital, okay, and mainly to maintain or in the dissemination of infectious or other harmful agents. This type of biosecurity, when you try to do it on animal premises, like even animal shelters facilities, or when you apply it in the clinical setting up, is much different and more challenges for us because of the several issues, but including the way we manage animals. And the animals is not like the issue with the biosecurity, is the people who are really dealing with these animals in that way. But biosecurity is the core of the preventive medicine, whether we deal with that in veterinary side or we deal with the human side. So we cannot, in my opinion, avoid dealing with that type. Biosecurity until recently was not really being identified. I can tell you is biosecurity, especially when it comes after September 11th, 2001, was not even used a lot outside of the, let's call it, production medicine and agriculture setting up. And mainly because of what happened with the biothreat and other issues, biosecurity start to be used, okay? And currently, it's really like it's increased that attention to be used and with the current pandemics that we are going through. I borrowed this slide from the slides I used in the training of program for biosecurity just we did recently in Central Asia and Southeast Asia as related to the African swine fever. And the reason I borrow it because to show is not really a major thing when we think about the term biosecurity. It's mainly a common sense into practice. And I listed all of the potential here for like biosecurity, particularly related to livestock premises in this situation. However, you could really replace that with any other like premises, including, let's say like nursing home or including like a daycare center or school. You could see how that can be applied as especially now when we deal with a disease such as or a virus such as the COVID-19 virus. And the reason I mentioned that because we have not really utilized what we know and what we applied in veterinary medicine in terms of to deal in the preventive measures that is needed for the COVID-19. And that's really, I would not blame it on anybody, but I would say, well, we have not established a good line of communication in that way. This slide, I want to emphasize there are three essential requirements or regarding the biosecurity for non-endemic agent and how it can be established. And these are, again, part of the slides I presented, as I said, in 2006 for the veterinarian at that time. Mainly, these elements is exclusion to maintain the pathogens outside of the premises. The second one is checking for the unexpected, mainly monitoring the health of the animals, and that way you can do it with the human in that respect. And the third one is the intervention, the preventive measures that is needed to be done and how you can have the plan. The most important part is neither of those will work individually. We have to have them in an interactive way in order to obtain the optimal biosecurity way. I will go through each of those with a couple of slides, mainly to elaborate on what I mean by these three elements and how is related to the current situation, whether with the COVID-19 or with some of the animal diseases that are threatening our food security and food stability in that respect. So to start with exclusion is there are some, I will call factual statement that we need to keep in our mind. Pathogens can move with animal or people. I usually say pathogens do not respect political borders. Pathogen does not respect the boundaries. So that's something we can try to understand. And zero risk does not exist. When we said like we can't exclude everything, that does not exist. That's maybe in books, but we cannot really do it in that way. Korea, or especially South Korea, is well aware about there is no such zero risk when it comes to certain diseases such as FMD, an outbreak that occurred a few years ago. In addition is movement of animals and equipment and people are essential for our operation, essential for maintaining our social interaction. This is what we are currently, especially politicians are challenged when you try to apply certain restrictions in the movement and people are complaining because it's interfering with our system, whether it's the trade or whether our social system or the quality of life in that respect. And another thing, relying upon quarantine or isolation is not usually adequate. Current COVID-19 experience in some countries in which, like they said, we close the border and we don't deal with things, or we quarantine those who will have the virus. It's not, I'm not saying it's not working, it's not adequate to constrain the virus in that respect. What we do for the measuring or the measures we use for the exclusion. The first and most important is screening, detection of the agent. And that's where also include early screening, early detection. That's part of what I mean. That's where a lot of issues related now to like testing, okay, for the COVID-19. And the same thing is for, let's say, African swine fever for veterinary medicine, is early detection. It will be very important in that way. The second one is we identify the source, where the infection or where the agents are coming from. So we can constrain that and maybe take action directly there. Another one is restriction in the trafficking, and mainly is separation. And that's where it come in a lot of ways by what happened now with the COVID, by reduction in a lot of trafficking. And a good example, what we are doing now, instead of going all together in Seoul, and I really would like to go there again, but I think is now, because of the constraint, we are trying to restrict this trafficking and being the separation, but maintain the communication in that way. Segregation of groups and movement restrictions. This is being used a lot in veterinary medicine. And when COVID came in the picture, some governments, and I don't want to mention like which countries, they started to segregate according to what they call high-risk group. And then not only restrict their movement, but they apply the early screening and early detection and try to at least treat these cases as much as possible. Again, is and I like to call it an issue like we have used before and in veterinary medicine. Measure for the exclusion, it depends on the overall sensitivity of the system. How we can exclude as early as possible. That's what it means. Not only the test, but the overall of the entire exclusion system. That means we need to have confidence in the exclusion measures, what we call in epidemiology the predicted values of negative, but not of what the test is, the overall of the system in that respect. And there are methods you can use in that way. but we should also have reasonable sensitivities. Avoid high percentage of what we call false alarm, okay? When we have like false positive, and we will call it in that way, because then you lose the faith in the exclusion measures. What we mean by the checking for the unexpected is mainly monitoring, and that could include production parameters. Okay, what currently is done with COVID, the monitoring is a good way, but maybe we need more indicators or more performance, such as how many they go to the hospital, how many they are vaccinated, how many they are like in certain age, and how many they are in certain profession and occupations, etc. So, and try to determine the health status on a population basis, whether in the animal population or in the human population in general. The benefit here is not only biosecurity, the benefit is the health status of the community in general. But that one requires resources and interest. And unfortunately, the monitoring will require comprehensive surveillance. Surveillance is not popular among the decision makers and the financial institution because they don't see tangible thing if the surveillance is working. Actually, success of the surveillance means is an effective of the surveillance means it's working, but also is not tangible in that way. How we do the measuring of the checking for unexpected, record keeping and good observations. To my surprise, okay, being in the epidemiology for more than 40 years, to my surprise, even in a country such as the United States, now we don't have a national record of who vaccinated for COVID and who is not. We don't have a national records like who got the disease and who did not. We don't have a national records how many tests being done in each individual and how many of them were positive and how many of them are negative. And as I said, I was astonished when I got all of these. This will require systematic collection of information, which is part of what I call national interest and part of the surveillance system. Will require maybe we do syndromic monitoring system, as we did with influenza in public health, as we do it in animal side, because fortunately, I said fortunately, our patients do not talk, so we have to depend on the syndrome, okay? So that's, again, is what we have done it in a lot of diseases in a very positive way. And we should not ignore the negative report. When we don't have things, that will be very important for the success of the monitoring system. And it requires notification process. It requires serious line of communication between both the providers and the user. And here I'm using the herds or the premises because that's what in 2006, that's what I presented in that respect. Now, the third element is the intervention. As I indicated about the intervention is mainly the preventive measures, but we need to plan for that. We need to be ready for the worst case, okay, scenario, and that's what is needed. It needs a plan in which be practical and executable, not only on paper, would need to be reasonable likelihood of success, and especially with rapid implementation. Today in the United States, being announced, there will be a rapid COVID test that will be available to get it from the grocery store. UK, for example, is available and is free. Once you request, it will come to you by mail in that way. And the reason I mentioned that, because that will give you an idea what's going on and allow people in such way to be responsible for monitoring what's going on and be part of the plan for the intervention or the prevention. At NICE, we keep a list of pathogens and disease with the possible intervention measures. With the COVID, for example, we should not only consider COVID-19 and influenza. Maybe we think about other viruses and bacteria, as now is being recognized how we are monitoring the foodborne diseases and how the foodborne diseases is going down. And now we need to be careful what does it mean in terms of that for the negative impact in the long time, because there will be some negative, in my opinion. Coordinate with other related industry and premises type. And I really think here, the intervention, like for COVID-19 now, what's the negative impact on both environment and the pathogen evolution if with the use of several disinfection that they are working beautifully to reduce the spread of the COVID virus. But what will happen to the, let's say, bacteria or other parasite? Are we building a new type of resistance? And if that happens, what are the options for the intervention that we need to take into consideration? What's the intervention measures? Usually we have vaccination, and we need to think, is it reasonable? And that's, I mean, I don't want to open that can of worms, because a lot of the discussion now about the vaccine and that way. Cleaning and disinfection. As I just mentioned about like the using of the disinfection for the COVIDs, cleaning and disinfection is being used in veterinary practice for a long time, okay? But also we have recognized how the cleaning and disinfection could also build resistance for some of the pathogens. We need to take that into consideration now for a human population under the One Health concept, how we can reduce the potential for the future of this type of issues. A treatment and use of mass treatment, okay, as preventive measures. Currently, and some of you hear about the pills to reduce the severity of the COVID-19, okay? And now there are some discussions, at least scientifically, that these pills should be available for whoever would like to have them, which in my opinion, it means mass treatment, okay? We in veterinary medicine, we learned a lot about the side effect, the negative on the environment from using mass treatment. So we need to address that issue if we try to use it in COVID-19. I know we are not destroying infected humans, but in veterinary side, destroying infected animals as preventive measures, okay, or as like a way to reduce the impact of the disease will have some negative environmental impact. And we need to really address the issue and look for options in that way. I have 2 more slides, and I think I will finish my talk. So what is needed for biosecurity and mainly to be proactive engagement in that way? We need to first participate in collection of baseline data. We need reliable information, not information to come through the social media. So that will require a comprehensive, customized checklist for every specific sector, okay? All the way from people working in different occupations to the animals that are in different situations. We need to have customized checklist. It can be used in that way. Another thing is we need to evaluate the impact on the environment assess before we use. And we need to be very careful to have these measures and any options for prevention to look for those before we go with that option. And another one is we need to explore alternative options for combining the biosecurity and the biosafety under one health concept. That's what I like to call total working environmental health. Okay, in that way, people working in like animal facility, for example, are part of the production unit of the animals that or that particular products. So we need to consider that in our assessment. And we need to work, and some of the previous speakers approach, and especially Dr. Lee, the Dean of the Public Health, indicated about various disciplines. We need to move from multiple discipline to interdisciplinary. I really think that's what we need to do, and think about the social well-being in a state of like thinking about protection of our discipline in that respect. And for that, I would like to end up my talk like we need to move away from what I like to call the silo, in which like education is in one silo, research in another silo, and the outreach and whatever program and in another silo. And then I'm including in the outreach is policy and action. What we need to move away from this silo by building a pipe between them, the interaction between these three silos together under one aspect in human health and the quality of life as our interest in general in that respect. That's where I also support what Dr. Lee was saying. Let's work on like what he called it, a school of One Health. And I really like to say like, let's work on One Health practitioners in a state of physician, veterinarian, environmental health, biologist. Let's work on that type of title in a state of that. And finally, I would like to say, I love this picture. We need to be very precise in our execution of a plan and be careful, okay? This father or grandfather, he doesn't want to lose the baby. So look what he's doing, but also he wants to be very careful in taking sharp picture. So here where I think is precaution, but also it's being precise and utilize the talents and the multiple tasks that we need to do. And thank you for listening to me. And I, as the organizer indicated, like maybe we don't have a lot of time for question and answer, but I am willing to respond to any question or rejection to through either my email or the chat for the, I will stay for a while before I will go for my dinner. Thanks again.
Okay, thank you, Dr. Salman. Oh, it was really insightful, and it was great for the first session, I think. And today he gave his presentation in the Scotland, not in US, because of his traveling schedule. So I hope you enjoy the dinner. Thank you, Dr. Salman. And as Dr. Salman indicated, he will be available for the chatting board and also email response. So if you have any question or comment or further discussion, probably you can use that tool. Now I want to introduce the next speaker, Dr. Sid Thakru, who is a professor of molecular epidemiology and director of the Global Health Program at the College of Veterinary Medicine, North Carolina State University. He was the associate director at the Comparative Medicine Institute and led the Emerging and Infectious Disease Research Program. Seed research is centered on drug-resistant bacterial strains. He will share some of the important issues regarding these topics. Please join me to welcome Dr. Thakru with big hands and smiling face.
Okay, thank you. Can you hear me?
Yeah, I can hear you. Thank you.
Great, great. Thank you for the introduction, Dr. Kim. Great to see you again. And I want to thank Dean Han and other colleagues and the organizing committee for inviting me to this wonderful colloquium. I really appreciate it. Professor here at the College of Veterinary Medicine, and what I'm going to talk about is the complexities really of antimicrobial resistance and why it is such a huge challenge. And if the challenge was simple, we would have solved it by now, but we have not. So what we really need is a One Health approach. So what I'm really going to talk about is, I'm going to start with my research background, talk a little bit about global health. You know, in the beginning, one of the speakers, Dean Han, I believe, was talking about, you know, collaboration and partnership. So as the leader of global health program at my college and my university, I would welcome my colleagues from Seoul National University to NC State, both for faculty and student exchange. So hopefully we can do that. Then I'm going to talk about antimicrobial resistance, AMR, from the One Health perspective, and why, you know, it's a huge challenge. And I'm just going to share a lot of data from my lab, you know, studies that we have been doing, studies that we have finished in the past. And I think with Dr. Salman giving a big picture, 50,000 feet view, maybe I'll come down a little further down and, you know, just share with what we are finding in the field and in the work we have done. So as a quick background, my research focus, I use a systems-based approach. I call it farm to genome system approach to focus on antimicrobial resistance. And this is a slide that I've not changed. I used this slide 14 years ago when I interviewed for my job at NC State, because the focus has not changed. It has been antimicrobial resistance using a One Health approach. What I have really done over the last 14 years is my focus on the environment has really become more and more in terms of where the challenges are. So we do a lot of sampling on farm, you know, both from humans, animals, retail meat. We bring the samples back, we test them in the lab, we isolate different bacterial foodborne pathogens that cause infections in humans and also in the animals, and have a huge reservoir in the environment. And then once you find the resistance, our major focus then is, you know, what are the genes that are causing resistance? What are the mechanisms of resistance? We do a lot of genomics. Of course, we also have a diagnostic aspect. But what I'm really interested in is the population and the global epidemiology. So I'm not interested in salmonella in the United States. What I'm really interested in is if the salmonella you find in the US, how similar or different is the salmonella that you would find in China or India, for example, especially in terms of resistance, because different countries have different restrictions for antimicrobial use. Many parts of the world, you can access antimicrobials without any prescription, and that causes selection pressure on these pathogens. And the same pathogens, they behave very differently based on selection pressure. So that's what I'm interested in, to understand how these emerging strains are emerging, what are the pressures that they're acting, and how quickly they move around. And I'll talk a lot about this in my presentation. My lab is home to 2 national surveillance programs. So they're both funded by NIH and FDA. The first one is the FDA's Genome Tracker program, where we do whole genome sequencing. We generate anywhere from 500 to 600 whole genome sequence profiles every year. And this is not just bacterial isolates from US, they're from all around the world. So that helps me in genomic comparison of these isolates from different parts of the world. And I also run the National Antimicrobial Resistance Monitoring System in North Carolina. So my lab generates the drug resistance profile for multiple bacterial pathogens from retail meat that people in the state of North Carolina buy. And that data goes into the FDA database. So these 2 surveillance programs are very important. And so really happy about that. My other hat that I wear is as the Director of Global Health Program for both the College of Veterinary Medicine and NC State. We are also the World Health Organization Collaborating Center in Antimicrobial Resistance and One Health, one of the 18 centers, collaborating centers that exist around the globe. This is the partnerships that the College of Veterinary Medicine faculty under the Global Health umbrella have. So these are real collaborations. We are in many different parts of the world. And I would really like to, bring South Korea as part of our partners. It'd be wonderful to have a collaboration, whether it's antimicrobial resistance or whether it's any other area. As a top veterinary school in Seoul National University, and as one of the top veterinary colleges in the United States, I would love to partner with you and discuss collaborative efforts that we can pursue. We heard about multidisciplinary, interdisciplinary approach. So that's what we use in our college. We have faculty with multiple backgrounds. I believe Dr. Yongbak Kim may recognize some of his faculty from the past. But we have mathematical modeling, transboundary diseases, molecular diagnostics, sequencing, AMR surveillance. So all these faculty, they work together in a collaborative fashion to address different global challenges that we are facing today in this world. We also have a very strong training program. We invite Fulbright scholars from different parts of the world. I was just looking at over the last 4 years, we have around 35 faculty, students from different parts of the world who come to our college and to our different labs for training. Again, I would welcome students from your university and your college to NC State. We also run a very robust global health certificate program for our DVM students, in which they have to do an 8-week experiential learning in different parts of the world. And it's been a very successful program. So we are really proud of it. Okay, so I'm not gonna start talking about antimicrobial resistance. We all know it's a huge problem. In the US alone, we see 35,000 deaths every year. It's a significant impact on animal health. On an average, we spend in the US around $8 billion every year to treat patients who end up in the hospitals with drug-resistant pathogens. And these patients typically tend to stay anywhere from 3 to 5 days more in the hospital simply because it takes more time to get rid of these drug-resistant pathogens. The challenge is, as I always say, we need global surveillance, and you can have all the surveillance programs and interventions you want in the United States or in developed nations, but this is a global problem because pathogens move around very fast. So just having nice surveillance programs in one part of the world is not going to solve the problem. And I think COVID is a great example. Pathogens don't respect international boundaries. And as long as they can move around, they are very happy. Drug resistance is a huge problem with no easy solutions. I tell people COVID will go away tomorrow, but drug resistance is here to stay. The ONI report in 2016 projects that by 2050, deaths caused by drug resistance will cross cancer. AMR pathogens will cause around 10 million deaths every year. There are no vaccines over here. And so Dr. Salman talked about biosecurity, which I will talk towards the end. I think those factors are going to be very important as we try and take care of this AMR challenge that we face every day. And AMR is a perfect One Health example. You have these pathogens, they infect humans, they infect animals, and they have a huge reservoir in the environment. And everything impacts these pathogens. So, climate change, increasing sea temperatures of the sea have been now attributed to increased transmission of drug-resistant pathogens. Okay, so environmental pollution, movement, trade, anything that can move, travel, anything that moves pathogens from one place to the other is a huge problem. So it's really, drug resistance is really a One Health problem, and it needs interdisciplinary approach, a One Health approach to find solutions. And there are no easy solutions at this stage. So that was my big picture view for AMR. And now I'm just gonna share a lot of data with you. Hopefully you'll find some interesting, and hopefully you'll find it challenging. So these are studies that have been going on in my lab for almost 10-15 years now. And I'm gonna just share with you the key findings of these studies. So we all know about drug-resistant Salmonella, especially the colistin-resistant E. coli. So colistin resistance, as we know, has been present in the chromosomes. So whenever these genes, they jump onto a plasmid, they cause huge problems. So the first mobile colistin resistant called MCR was reported from China in 2016. They isolated that from a pig, an E. coli. But then we were doing this study, working collaboratively with the state public health lab, and we had salmonella isolates from 2014. And one of my DVM students, Valerie Nelson, who was working on this particular study as part of a summer project, we found out that 2016 was the first report of MCR1 from China. And then we found out that we had a isolate the Salmonella enterica 452I- that carried this mobile colistin resistant gene. And that was isolated from a female, an 18-year-old female who had a travel history to China in 2014. So she had gone to China in 2014, and then she came back to the US, and within a week of coming back to the US, within 2 days of coming back to the US, she fell sick, she was hospitalized. And from there, we isolated this MCR 3.1. So the point I want to stress is this MCR 3.1 was already circulating even before 2016. Just because it was detected in 2016 for the first time doesn't mean that that's the first time it emerged. So again, tells you that travel really spreads it fast. So when you look at the plasmids that are existing in this particular serotype of Salmonella, you can see it 100% matches with the MCR 3.1 that was reported from the pig feces. Then we also found out through some more research that there was a Canadian patient with a travel history to Thailand in 2013 who had a similar MCR 3.2. The only difference was a slight deletion and around, it was almost 95% matching with what we have found over here. So the point is, pathogens travel fast, they go around the world very quickly, and once the genes move on to a plasmid, the vertical and horizontal transmission just increases significantly. So one of the solutions that have been proposed for drug resistance is to take out antimicrobial use from growth promotion purposes, which has happened now in the European Union, and it's also happened now in the US. So one of the studies we did, a USDA funded study, was to see what happens when you compare indoor pigs. So these are pigs that are commercially raised, and that time they were exposed to antimicrobials, and compare them to pigs that are raised outdoors. So these are backyard outdoor pigs that are not given any antibiotics, and they have the antibiotic free tag that they have. So if you follow the normal hypotheses, indoor pigs, since they're exposed to antibiotics, should share drug resistant pathogens, and outdoor pigs that are not given any antibiotics should share drug susceptible pathogens. We looked at multiple pathogens from these two different pigs. We did a lot of environmental sampling also. This is around 2012. I really started getting interested in what is the role of environment in transmission of these pathogens. So a 5-year study, but I'm just going to wrap everything in 2 slides. So this is looking at Campylobacter. This is the pig population. This is the environment population over here. And the green bars are the resistance that we detected in outdoor pigs. So these are pigs not given antibiotics, and the red bars are the resistance to pigs that were given antibiotics. And here are the different antimicrobials listed over here. We use broth microdilution. So this is the pig, this is the environment. And if you just look at both of them, the impression you get is what is inside the pig is what is in the environment. So it's almost a mirror image. So what we are finding in the environment was exactly what was inside the pig. And we did find a lot of resistance, even in pigs that were not given any antimicrobials. So what was the reason? Why were pigs who were not given antibiotics still shedding so many drug-resistant Campylobacter strains? So when you do the phylogenetic analysis using multi-locus sequence typing, what we found out at the end, when you look at the unique sequence types, is that it's actually the same population of Campylobacter that is dispersed in both the pig populations. So it does not matter whether you were given antibiotics to one pig and antibiotics not to the other pigs. Since the same population of Campylobacter is spread or disseminated in both the populations, that's why we find a lot of resistance. So why do we have the same population of Campylobacter spread in both the pig populations? There could be many reasons for that. North Carolina being close to the coastal hurricanes have always been a big problem. And that could be one reason why the dissemination of these drug-resistant pathogens has happened across both the production systems. So again, the point I want to stress is, even if you don't give antibiotics, does not mean that you will not find resistance in that animal. The other thing I want to stress over here is something called ecological niche. So when we just go up and start talking about pathogens, we have to also understand what is their fitness and what is the dominance based on the environment that they exist. So in the same study, when you look at Campylobacter inside the pig, these red dots are indicating a specific population of Campylobacter coli in the pigs. When the same pigs, they go to a slaughter plant for processing, they have to, by USDA mandated, they have to rest in a big resting area called the larage. Over here, we have a different population of Campylobacter that's very dominant. And what happens is, in a matter of couple of hours, this dominant slaughter population of Campylobacter completely wipes out the farm population, to the extent that what you find in the finished product is what is existing in the slaughter plant population. So again, just tells you how dominant this population is, and this competition that goes on between these different populations, even though they are both Campylobacter. In the same pigs, we have seen, and this is the data we have for almost 10 years now, is typhimurium. So if you just look at typhimurium and say, oh, I'm just dealing with typhimurium, but we always say, okay, if you're talking about typhimurium, which specific phage type of typhimurium you're talking about? So this is phage type DT104. It is resistant to 5 antimicrobials, and all the genes are located on a class 1 integron, which is stably integrated into the chromosome. When you look at another phage type of typhimurium called DT104, which is always found on the floor, it is resistant to only 3 antimicrobials. The ampicillin and the chloramphenicol genes are lost. And what is really interesting, I call it ecological niche. What is really interesting is that the population of Salmonella typhimurium DT104 inside the pig and the one on the floor, they don't cross each other. So the pig, inside the pig typhimurium, you will not find on the floor. The one that's on the floor, you will not find inside the pig. So again, these populations are constantly in competition with each other. And it's very important to have an understanding of what sort of population is existing inside the pig and outside to have real strong interventions. Otherwise, interventions are not going to work. So then we are moving towards something called organic system. You know about the organic system. So, USDA mandated, no antimicrobials for treatment, no disinfectants in the environment. And this is a USDA NIFA funded study for $10 million, has got multiple universities as part of it. So we are looking at poultry in this case, again, focusing on Campylobacter, Salmonella, and extended spectrum beta-lactam E. coli. And the idea is to not just look at the pathogens inside the bird and what's in the environment, but also look at the microbiome and to see how the microbiome is actually guiding what sort of population is existing in these birds. And I'm just going to show you a couple of slides over here. So why it is important, if you look at the US, at the CDC right now, there's a big outbreak of Salmonella that's going on because of attributed to backyard poultry. And it's a big problem. It's still ongoing. It is right now, this is as of a week of August 31st, 863 cases, 2 deaths, 209 hospitalizations. And this is spread in 47 states. It's really widespread. So that's why what we are doing in this backyard poultry is very timely. So this is an ongoing study. I'm just going to share one slide with you on extended spectrum beta-lactam E. coli. So these are birds that have no exposure to antimicrobials at any stage, but you can see, I'm showing you a very preliminary data with 51 isolates of ESB E. coli, and the red bars indicate resistance. And you can see there are a lot of red bars. And again, you would imagine, how are these birds getting exposed to an extended spectrum beta-lactamase, which is really a very a pathogen that you don't want to get infected in because it's resistant to multiple antimicrobials. And we are also finding resistance to ciprofloxacin. And this is what we see. A commensal like an E. coli starts picking up these ciprofloxacin resistant genes, and then they start shuttling around to other enterobacteriaceae like salmonellas and shigellas, and that's when it becomes a huge problem. So again, we are finding these in the birds, we are finding it in the environment. We are now doing microbiome focused studies to understand why this is happening. But it is happening and people are getting exposed to it. So why are we finding resistant pathogens in systems that have not been used, that have not been exposed to antibiotics? So this is the data from fluoroquinolone resistant Campylobacter jejuni from humans and poultry in the US. So in the US, fluoroquinolones were banned for use in poultry production in around 2005 and 6 because the number of cases in humans were going up that were in Campylobacteriosis that were resistant to fluoroquinolones. But you can still look at the data after 2005. Resistance in poultry meat to fluoroquinolones has not gone down. It is still there. So you can look at the Denmark data also. Denmark has been the leader in not using antimicrobials for growth promotion for almost 20 years now. They banned everything in the year 2000. But this is the latest data for 2019. And you can look at ciprofloxacin resistant Campylobacter from broilers and cattle. And you can look at this. The resistance in broilers is 45%. And when the same birds, they go to a processing plant and get transformed into broiler meat, the resistance actually goes up to 65%, which is very high for a country that does not use any antimicrobials for growth promotion at all over the last 20 years. So why do we still have resistance floating in there? The hypothesis should be if you don't use antimicrobials, resistance would have gone away. And the reason is very simple. In Campylobacter, fluoroquinolone resistant Campylobacter is just a better fit strain of Campylobacter as compared to the susceptible strain of fluoroquinolone. A very simple study that was done. You can see when you give birds fluoroquinolone, when you challenge birds with sensitive Campylobacter jejuni, next day you will find only susceptible Campylobacter jejuni. You give another group of birds resistant Campylobacter, next day you're going to find resistant Campylobacter in the fecal sample. When you mix both of them together, you're going to find only the resistant strains of Campylobacter jejuni in the fecal samples the next day. So competitive exclusion or fighting between these resistant and susceptible strains, and the fluoroquinolone resistant strain is just a better fit. So even though you don't give any antimicrobials, because the fitness of these strains has improved so much, they tend to stick around for a very long time. So I showed you the example of Denmark, where in the farm the resistance to ciprofloxacin was 45%. When the same birds go to a processing plant, the resistance to fluoroquinolone went up to almost 65%. So there is something happening in the environment within the farm, within the processing plant, that the resistance goes up. So this was a study we published last year in PLOS One, where we looked at the entire state of North Carolina and the US in terms of farms, in terms of processing plants, and in terms of retail meat. And we found some very interesting data. So we got data from USDA FSIS for farms, from APHIS for processing, and the retail sampling we are doing in my lab anyways. We isolated Campylobacter, and then we have a genomics, bioinformatics genomics pipeline that we use to figure out what resistant genes are and what sort of sequence types are existing in these system. So let me just quickly explain to you what a sequence type is. So and what a clone complex is. So the basic thought process is all the genomes, they originate from what is called the founding genotype, and that's called a sequence type 1. And these founding genotype, because of variations in the genome, Campylobacter, for example, has a hypervariable genome. That means they like to talk to each other. And so they accumulate these genomic changes, and they soon diverge from the founding genotype, and now they're called single locus variants, these single ST2s. And the single locus variants over a period of time will accumulate some more genome inside, will make some more changes, will further diversify, and they're now called double locus variants. And soon you will have triple locus variants. But as long as they are not that spread out, they are still part of the clone complex. So in Campylobacter, for example, coli sequence type 828 clone complex is the dominant one. So what did we find? So you can see a lot of number. These are sequence types. So these are unique sequence types that were detected on the farm, in the processing, and in retail. And just by this, so every sequence type is a unique Campylobacter jejuni or a coli. And one thing that you can see from this particular slide is that there is a lot more numbers in the processing plant. And the reason we have way more numbers in the processing plant is because birds are coming from different farms and they accumulate in a single processing plant. And so they bring the unique genotypes from their own farm environment and they sort of dump it into the processing plant. That's why there's a big influx of sequence type. But what is really interesting is once they get cleaned up and they go to the retail, all the Campylobacter coli is gone. All you find in the retail meat is actually Campylobacter jejuni. And also that is reflected in 95% of the infections in humans is caused by jejuni, around 5 to 10% are attributed to coli. So again, a fascinating example of survival of these unique strains of Campylobacter depending on the environment. And when you put them all together, you can look at what is happening over here. So this is a very interesting slide. So Campylobacter coli and jejuni, because they're hypervariable genomes, they talk to each other, they exchange genomic material. And here is the key challenge to keep in mind. On an average, Campylobacter coli is resistant to more number of antimicrobials, and Campylobacter jejuni causes more number of infections in humans. But imagine if they start exchanging the genomic material to the extent that many of the resistance mechanisms that you find in coli jump into jejuni, and all of a sudden you have this super pathogenic Campylobacter jejuni, which not only causes infections in humans, but is also resistant to more number of antimicrobials. And this is a slide that shows you, when you plot the sequence type, you can see that Campylobacter coli is over here and the jejuni population is over here, but there is right in this middle is where the Campylobacter coli and jejuni are merging with each other because they have the same GLTA gene, which is one of the housekeeping genes. So this is the situation that happens when pathogens come and talk to each other. And if they have a hypervariable genome, then they're going to come and blend to each other and they cause more infections and more problems. So that's why it's very important to use not just a very one approach to target antimicrobial resistance, but to look at all the way from 50,000 feet level and bringing it down to 10,000 feet level to really understand what the problem is. So the last study that I'm going to share with you, this is a big comprehensive study that is under review right now in antimicrobial agents and chemotherapy. It's led by my colleague Arnaud Van Vliet in the School of Veterinary Medicine in University of Surrey in the UK, where we have compared more than 40,000 whole genome sequence profiles for Campylobacter coli and jejuni. So we are doing countrywide comparison between US and UK over an 18-year period. So over 18 years, we have tremendous amount of data. It took us a lot of time to analyze it, but I'm just going to share 2 more slides with you as to what we are finding. So this is Campylobacter coli, this is jejuni. Obviously more jejuni isolates than we have for coli from, and we have different antimicrobials listed over here. And the sources are farm animals, humans, and poultry. And in the blue, we have UK, and the red, we have the US. And you can see some differences between colis and between jejunis across the 2 different countries. So tetracycline resistance, I tell people to stop looking for tetracycline resistance because practically all the different pathogens are now resistant to tetracycline. It's so spread out. But the interesting ones are macrolides and fluoroquinolones, and of course, aminoglycosides. So you can look at the differences you can see in the US, for example, poultry aminoglycoside resistance was higher as compared to the UK. But on the UK side in farm animals, so this is cattle and other animals that you can think of, pigs, you see higher resistance to macrolides as compared to what you find in the US. But the key one is fluoroquinolone. Okay, and fluoroquinolone in poultry in the UK is significantly higher than what you find in the US in coli. And in jejunis also, you can see the same thing. Fluoroquinolone resistance in humans and also in poultry is significantly higher than the US. Macrolide resistance is very low. Macrolide resistance also is much a factor for coli than for jejunis. So if you compare the data, the big picture for us is the prevalence of resistance has not gone down over time. These are one of the 2 leading countries to try strong control on antimicrobial use in food animal production, but the resistance has still has not gone down. We can look at from aminoglycosides, for macrolides, for quinolones, and for tetracycline, for coli's and jejunites. And you can see the resistance is hovering for some down the lane, but still the resistance to important ones like fluoroquinolones has not gone down. Okay, and the reason for that is the fitness of these strains, the presence, the multiple mechanisms of resistance, and a very heavy reservoir of these resistant mechanisms and these genes in the environment. So antimicrobial stewardship alone is not effective, is the big conclusion for us. And what Dr. Salman was talking about, biosecurity is very important over here. Simply taking off antimicrobials is not going to solve the problem. Biosecurity is very important. And controlling these pathogens, you have to have very smart, strategic ways to understand it and to not take them lightly. I think the challenge is we think all salmonellas are the same, all campylobacters are the same, all E. coli are the same, but they are not. They are very different from each other, depending on the species, depending on the serotypes, depending on the host, and they behave very different from each other. So that's why it's a complicated problem. That's why we need unique solutions, piecemeal solutions to solve this problem. Judicious use of antimicrobials, strict biosecurity measures, those are some of the things that we need to do to really control it. So a One Health approach is very important. You can't do it without it. So to summarize, AMR challenge requires a One Health approach. As I've shown you, it's a very complex topic. There are no silver bullets. We have tried not using antimicrobials. There are some good results for sure, but also resistance to other antimicrobials has continued to go up. And a One Health approach with physicians, veterinarians, food safety experts, environmental sciences experts, social scientists are very important. We need an interdisciplinary approach. The previous speaker, Dr. Salman, also talked about this. Pathogens don't respect international boundaries, and the role of environment is very important. I'm very happy to see that you have another session after this, just focusing on the environment. I'd love to hear what the speakers have to say. And we have to invest more in education, research, and engagement. Education is very important. AMR, as I showed you, is a complex entity. No easy solutions, complexity surrounding it, ins and outs, and education is very important. And I say again, strict biosecurity, more vaccines are really good. At the end, I will say AMR is a global problem and it needs global solutions. So I want to thank my funding agencies again, and thank you so much for your time, Dr. Kim and Dean Han. Thanks for inviting me. I really appreciate it. I don't know if you have time for more questions, but I'm going to be here to answer any questions later on. Thank you.
Okay, great. Thank you. Thank you, Sid, for the informative presentation and also especially sharing your unpublished data. I do appreciate. And I mean, it is really glad to have an old friends, even through the online system. Hopefully we can see in-face meeting after the pandemic season. And also, I mean, as Sid mentioned, we can expand our educational collaboration or collaboration on the research program as well. So again, thanks, Sid. Okay, it's time to have another speaker. For the last speaker for the session one, Dr. Shuhua Wang, who is an infectious disease physician scientist and professor of medicine at the Ohio State University in Columbus. She is the director of research and implementation science for OSU Global One Health Initiative. Dr. Wang's clinical research interests include the diagnosis and treatment of tuberculosis in human and animal, using One Health approach in China, Ethiopia, Guatemala, and the US. Please join me. Welcome Dr. Wang with big hands and smiling face.
Thank you so much. Can you hear me okay?
Sure, yeah.
Thanks. So I want to thank Dean Han, Vice President Choi, and Dean Lee, as well as Dr. Kim for this wonderful invitation. So truly blessing to be able to present to the worldwide audience, especially in Korea. I was actually born in Korea, so it's very touching for me. So today I'm actually going to step back a little bit and kind of talk about Global One Health Initiative, which is our organization, and how our Ohio State University, our mission, and how we were able to accomplish some Global One Health. I really appreciate the presentation given by Dr. Mo as well as Dr. Sid to give the big picture of Global One Health and then the more specific. And now I would like to kind of focus a little bit on our education. So thank you again for the wonderful introduction. So just to give some people some background, where is OSU or Ohio State or Columbus? So we are kind of where that circle is, and we're about 5 hours driving to Chicago, 8 hours to New York. So not too far from everything, but we are very much known for our OHIO. If you ever come to OSU, I will invite you to a football game and everybody will be chanting this. And everywhere we go, we like to take our pictures with our OHIO. So you can see this. And then we do have beautiful skyline. And for me, this OHIO could really be One Health in Ohio, right? So again, OHIO. So I don't want to belabor this point because I think it was so well spoken by Dr. Mo and Dr. Sid. So really One Health is this collaborative, disciplinary kind of goal where we're really bringing human, environmental, animal, and plant all kind of together. And as we could see, there's many different aspects that are involved in this, including zoonotic diseases, comparative medicine, antimicrobial resistance, food safety, security, environmental health, and even non-communicable diseases and cancer. So just stepping back a little bit more at this global risk landscape, we could certainly see that climate action failure, extreme weather, biodiversity loss, water crisis, infectious disease, food crisis are all contributing. And if we don't think about it in this Global One Health manner, and we don't want to be siloed, as Dr. Mo kind of said, then we're not going to be able to solve or achieve this problem. So just kind of looking at the planet that we lived in today and in the future, you can kind of see this African bulge as it's stated in this thing. So there's 7.6 billion human population, and by 2050, 10 billion people. Clearly, there is an explosion of human. And where can we really join this capacity to tackle complex global problems? We really need to really think about who is going to be out there doing this. And we can certainly see with SARS-CoV-2 that a pathogen in one area of the globe can really be transmitted all over in a very short time frame. And we can also see that there's many infectious pathogens are from the zoonotic origin. So again, a One Health approach is very much needed. And even when we look at just a bowl of salad, there's more than products from 10 countries that are contributing to this salad bowl that we eat. So if we're trying to track down a potential pathogen or an illness with outbreaks, then we are going to have to be able to do this. And as Dr. Sid had mentioned, the antimicrobial resistance is going to be a major factor by 2050, more than 10 million human deaths per year. So we need to get on top of this now. So the Sustainable Development Goals, there are 17 different major things. So how are we going to be able to do this? I feel, and we feel, that academia, so universities such as yours, are the key players in this, because only in this sector are we able to really align ourselves with 17 of these SDGs. We don't need to do every single one because we don't do every single one very good. But if you take a look at the global picture, how do we exactly fit in? What is our competitive advantage, and where can we make mutually beneficial, sustainable impacts? So really academia is a key factor. So I really want to kind of talk about, and I think everybody wants to invite you to come to our institution, and OSU is just as welcoming. So hopefully post-pandemic, you guys can come. But if we look at the student exchange, academic mobility, we can see that 32% of the US students that study abroad, they really go to these key countries, UK, Italy, Spain. So these are really the developed countries. And where is a global problem? Yes, we have it in UK, Spain, but the majority need are elsewhere. And if we look at the people coming to the US, 50% of international students are from China or India. But again, where do we really need some of these sustained educational maintenance. And so that is kind of like one of our mission as Global Health Initiative, is really to expand the capacity. We want to build workforce capacity. We want to train individuals in the country so that you can sustain, and then you can implement research, you can do surveillance, you can have outreach extension, resource stewardship, and financial sustainability. That is really Global One Health Initiative, or OSU Go Heights mission and vision. So I want to kind of step back a little bit more to just kind of tell you how we started. So OSU is a perfect campus and very similar to Colorado State and NC, where there's multiple diverse units or colleges within one campus, so we can collaborate. We have more than 20 OSU units, 7 health sciences colleges, food and agriculture, environment, business, engineering, social sciences, education, and human ecology. And as part of this Global One Health, we do participate in more than 13 colleges. And you can kind of see by this map down at the bottom how everyone is kind of interlinked. And Go Ahead history, we're actually quite young. As you can see by my backdrop, we are celebrating our 10th year. So we've really been in existence for 10th year, but you will be really impressed by the impact that we have made. And so it really started with kind of an idea in 2007 with an African pilot project. In 2009, we were able to receive some funding and some grassroots efforts. We really began to have a formal establishment in 2012 with the One Health Summer Task Force, where we do a lot of our education. And then since then, we have had an exponential growth. So again, we're celebrating our 10th year, and we have been having kickoff since September, culminating in November with World Health Day. So we have really been able to connect, convene, catalyze, and conduct our research as well as our training, really the key in globally all around the world. And we hope to include South Korea as well. So in terms of our experimental and transformational academic experience, it's really a unique experience where we try to engage students, faculty, and again, to go to the country to see what is needed and to really make that impactful program over there. We do have projects, certificate programs for undergraduate, graduate training. And then we really want to strengthen this global research coalition with faculty opportunity to gain additional funding, to provide opportunity for students and faculty. We want to be able to capture our impacts in journals, as well as publications, and to be able to integrate communities and influence health policies, where we know that even if we do all these implementation science, without the policy implementation, it's really difficult to make that high impact. And again, global doesn't just mean international, it means local, national, as well as global. So here I am in China doing bovine tuberculosis research. And then the bottom picture, it's just in our Ohio, Kentucky, West Virginia, Appalachian projects. And we really did have a great opportunity with SARS-CoV-2. We were already on the ground in Ethiopia, and within a few weeks of the initial outbreak in 2020, we were able to train 77 healthcare workers who could do surveillance and be disseminated to 8 different regions. We helped with Ethiopia in terms of their SARS-CoV serial surveillance. We're currently helping them to build capacity both in workforce and laboratory. We were able to ship supplies and equipment over in a very short period of time. We conducted NPI, or non-pharmaceutical intervention surveys, where we survey more than 800 individuals at different time points to gain their knowledge, assessment, and attitude towards SARS-CoV and behaviors. And then we were able to make implementation and recommendations to the Ministry of Health, where those immediate measures were taken for mitigation efforts. So again, very high impact. And so we want to be able to thank and appreciate all of the partners that we have been able to connect with in this last 10 plus years. And I want to just kind of talk a little bit about some of the projects that we've been able to do to really integrate this global One Health approach to be able to build capacity and mitigate disease and the human, animal, planet, environmental. And again, it is very challenging and very difficult. So there is a very close connection between human and animal, as it has been stated. So in terms of our main backbone of education is our Summer Institute. We used to travel to Ethiopia and train a lot of people on site. And these last 2 years with the COVID, we have really maximized our webinar models and really increase the capacity by which we could do training. So these are still a little bit older numbers where we trained more than 1,000 people from multiple countries. And again, these numbers will really escalate this year when we calculate our numbers. And then when we were there on the ground, we not only tried to do some of the general teachings in terms of the medical, veterinary, molecular genotyping, but we also want to empower them to be able to write grants, publish. And so there's a lot of hands-on workshops for them as well. And again, all of these have been transitioned to web-based, and we actually have them on recording so that they could be disseminated. So again, training capacity is the strength of our core. We want to be mutually beneficial for Ohio State as well as the global health community. We, excuse me, founded on current and future complex issues with open door research policy and sustainability. And as always, funding is a limitation in many parts. We try to provide some funding, but it is very difficult to bring someone over here for entire training. And I want to talk about our FOGAted training program. But before that, we have a very successful twinning program. So this is actually a funded program with the World Organization, OIE, which is the animal side of the WHO, as well as OSU and university. So it's headed by one of our veterinary, Dr. Armando Hoy, where we actually take veterinary students from Ethiopia or other countries, bring them to OSU, where they come to OSU Veterinary School, and they're actually taught one-on-one with other students. And then our students go over there and then they also participate. So it is truly a twinning program. And this model actually has really been disseminated, again, with the blessing of OIE. So hopefully there could be potential exchanges with your university as well. So very, very interesting. The next one is our NIH, National Institute of Health R21, or electronic capacity building. So this really has helped us launch our electronic platform where we can reach a much more large audience. We have a platform by which our instructions and different things are also logged so that people could tune in. And we're currently in a transition phase, but we will be able to have that up and running very soon. And then we have our monthly webinar, which is a Global One Health webinar. We have international as well as national speakers, and we have a site, and you're more than welcome to join in. We have multiple different speakers. And then we have this biannual meeting that has started more than 10 years ago, started in Ethiopia. Every other year we go to a different country. The other years are in the US. So we've gone to Ethiopia, Brazil, Thailand, Qatar, and Canada. This last year, we were supposed to go to France. However, with COVID, it has gone completely virtual. And most of us that does a lot of global health, at least for me, my passion is to travel. So I love being part of this community that can really travel. So hopefully maybe South Korea could be one year. So just to kind of go back and step back, and as I talked a little bit more about some of the projects that we were fortunate to be awarded and how we're really building the capacity. So very early on, we received a Global Health Security Award from the US Center for Disease Control and Prevention. And again, really looking at a lot of One Health matters. So rabies, it is a major killer that is preventable amongst children, where there's more than 60,000 deaths. So we really started this as a grassroots. We went to a smaller city outside in the outskirts, and then where rabies was a problem, where they looked at the dogs and what exactly was causing this problem. And then we trained and we vaccinated, and we continued to establish a national policy. We participated, and currently there's actually more than 100,000 dogs and cats that are vaccinated in Ethiopia, and we continue to participate and work with the CDC on this project. So very successful, and we have made a lot of educational campaigns at different levels. Brucellosis is another One Health approach where you actually have human as well as animal components. So we were part of this project where we went to 3 different areas within Ethiopia. We collected more than 800 human samples, as well as 800 samples from cows, sheep for blood and milk. And we were doing surveillance testing. We actually train Ethiopians in the Ethiopian Institute for Human, as well as in the national animal sector to train. And we built the capacity for the region. And so we were actually able to look at the serological prevalence. So we've definitely found that in one area, especially in the South, where the practice was more where there was closer proximity between human and animal and drinking of blood products, the serosurveillance was high. So a lot of education is to follow. And that Brucellosis project has completed. We're currently working to transition to anthrax. Antimicrobial resistance, as Dr. Sid had talked about, is going to be a major player in the years to come. And we actually have been doing antimicrobial training in the human sector to really train people how to collect specimens. And what our laboratorian always says, if you give me garbage in, garbage out. So unless we can collect the correct specimens and do it in a sterile fashion, we're not going to be able to get those antimicrobial susceptibility testing. We're not going to be able to provide the clinicians and the veterinarian with the correct pathogen sample so that they can actually treat correctly. So we have developed this webinar as well as a manual that has been disseminated in East Africa. And then we're continuing to work with CDC on antimicrobial resistance. And lastly, leadership capacity training. We really feel that we need to empower the local leaders in order for them to take charge. So again, our goal is really to increase capacity for workforce as well as laboratory. And so that is a key player for us. Other things that we're so proud of is our D43 or our NIH training grant. Is really a sandwich program where we have students from East Africa, Tanzania, Ethiopia, and Kenya, where they're part of a PhD program that they have already been enrolled in. Then we bring them to OSU for 1 year. Most of them want to do molecular training because that technique may not be available for them in their home country. So they do that for a year. They learn to build capacity with a US mentor as well as their local advisor. And then we send them back and we continue to help them gain as they increase and complete their training as well as build their academic career trajectory. So again, this is in molecular epidemiology of food, water, and vector-borne diseases, a One Health product. So these are some of our D43 graduates that have been and very successful and are current leaders within their own institutions. And many of them were actually so well trained that they have been the key leaders in SARS-CoV-2 for their own countries. So you could see many of our graduates and their program that they have been involved in, and additional graduates from our program. So we have continued our training program, and Dr. Sid had talked about this genome tracker for Salmonella. So we actually, because we have so many students from all over the world, we're actually continuing to build their capacity. And so they are sending isolates to this genome tracker, and we're continuing to work with them. So again, this is available to you as well. So anyone, anywhere can be submitting these data. And then we have leveraged other partnerships. So we're part of the Chan Zuckerberger initiative, where they actually came on board and really trained our fellows, the D43 graduates. They actually went to Ethiopia and Kenya to do additional training. And again, these type of training are what is really lacking in these countries. So we continue to work on implementation science. We're part of a large USAID tuberculosis trial. We just completed and published our findings as part of 3 sites, Ethiopia, Mozambique, and South Africa. We contributed more than 800 enrolled HIV/TB patients in a randomized controlled trial. And this has just been published in the annals. So other really wonderful project that have continued to go on is Campylobacter genomic and enteric dysfunction. Again, looking at childhood stunting in terms of the Campylobacter infection, so another One Health, and really working with other partners, University of Florida, as well as the Bill and Melinda Gates Foundation. And then Tartar is our food safety that was funded by Bill and Melinda Gates, looking at food and dairy focus, really on different pathogens that are enteric, and then risk assessment. So a lot of projects have been on hold due to COVID. So we have learned that they have just gone into the field to start collection. So we're really hopeful that this will progress quickly. And then it's also training, again, PhD fellows on the ground. So this is one of our projects, again, led by Dr. Armando Hoyt, who has an antimicrobial resistance project. He was the one who was doing the twinning. He actually has a Central and South America project in terms of antimicrobial resistance. Again, another one of these training on-site projects with 14 Caribbean countries, 33 participants looking at antimicrobial use as well as resistance in food safety, public health, regulatory issues. Again, policy change to make the thing. So we're really trying to improve our impact statistics because we could do all this work and without recording, we're not going to be able to know. So we have a monitoring evaluation team that has been brought on board to really help us capture some of these impactful things. So the time really is now for academics, so institutions such as OSU and yourself, to really take charge, opportunity to be the leader in Global One Health, to have the return on investment, to have return on mission, preventive diseases and education leaders advance, converge impactful research. Again, enhancing your and our university's visibility and reputation. So this is my thanks and my final slide. This is Dr. Francis Collin, Director of NIH, and Dr. Roger Glass with the Fogarty Training Grant. And bottom is our NIH Gold Award that we were recently given. So again, thank you so much for this opportunity to present. I'm happy to take some questions, and we really want to continue this global education and capacity building. Thank you.
Okay, thank you, Dr. Wang, for the great presentation. And also you shared a lot of activities in your institution. It was really, really impressive. Again, I do appreciate Dr. Salman, Dr. Thakru, and Dr. Wang for your great presentation. I know you had a very busy day, and now you gave us presentation at night, sacrificing your off time. And please give them big hands for their wonderful contribution. With this, first session is closed, and the second session with the theme of environment and One Health, and it will begin on 10:30 a.m. Thank you, and see you all in the second session after the break. Thank you.
Thank you so much.